Hello everyone, I'm Kumar Gaurav, a postdoc researcher at the John Innes Center. I will be talking about the genetic diversity of Aegilops tauschii, the D-genome progenitor of red wheat, how it contributed to the wheat evolution, and how we can exploit it to expand the existing wheat breeding pool. Hexaploid wheat originated from the hybridization of tetraploid wheat, Triticum turgidum, with Aegilops tauschii. This event sampled only a tiny fraction of the progenitor's diversity. I'm going to present our recent work, which provides further insight into this evolutionary process. Genetic relationship between Aegilops tauschii and wheat D-genome is important to understand the wheat origin and its subsequent evolution. In the previous studies, it has been reported that Aegilops tauschii is divided into two evolutionary lineages, lineage 1 and lineage 2. The phylogenetic analysis of Aegilops tauschii and wheat D-genome grouped wheat samples into a narrow clade close to lineage 2. It has been reported in the previous studies that lineage 2 is the major contributor to the wheat D-genome, with minor contribution from lineage 1. Here, we are reporting for the first time the discovery of a new basal lineage of Aegilops tauschii, which we call lineage 3. Accessions belonging to lineage 3 were earlier thought to have evolved from hybridization of lineage 1 and lineage 2. We conducted reference unbiased analysis using k-mers with an even sampling of 3 groups. From the results of the structure analysis, lineage 3 does not seem to have evolved from an interlineage hybrid. Instead, it seems to be a basal split from lineage 1 and lineage 2. This interpretation is further supported by the PCA analysis and the F statistics, which show that the 3 lineages are roughly equidistant from each other. To represent wheat, we added Chinese Spring D genome in the structure and PCA analysis, where it is grouped with lineage 2. However, in the structure analysis, Chinese Spring D genome is found to have around 2% admixture of lineage 3. This deserves a closer look since such an admixture has never been reported before. We need to make sure that this is not a statistical artifact. We looked closely at the signature of the 3 Aegilops tauschii lineages in 2 wheat lines, Chinese Spring and Arena, whose reference genomes are available. For each segment of the D genome of these 2 lines, we determined which lineage had predominant genetic contribution to that segment. The widespread presence of red color indicates that lineage 2 is the major contributor to the wheat D genome, but it is also clear that there is a non-trivial contribution from the other 2 lineages. In particular, the signatures from lineage 3 and 1 are present towards the telomeric regions of all the D genome chromosomes. Could it be that the Aegilops tauschii sample, which was involved in the polyploidization event, was itself an admixture of the 3 lineages? This seems unlikely because in many cases, Chinese Spring and Arena have alternating lineage 2 and lineage 3 alleles at the same locus. To explain this, we propose the multiple hybridization hypothesis. According to this hypothesis, the extant wheat originated from the hybridization of tetraploid wheat with lineage 2 of Aegilops tauschii. At some later time, another hybridization event between the tetraploid wheat and the lineage 3 took place. The 2 streams then recombined to give rise to the existing hexaploid wheat, having the varying admixture of different Aegilops tauschii lineages. So far, we have been talking about the Aegilops tauschii diversity historically captured in wheat. Next, I will present how to tap the diversity which was not historically captured. We do this by sequencing the Aegilops tauschii panel using k-mer-based GWAS to clone genes for desirable traits and deploying them in hexaploid wheat. Here are some phenotypes for which we explored the variation in the Aegilops tauschii panel. The first is powdery mildew resistance, for which we get a discrete peak on 7DS using k-mer-based GWAS. In the interval of around 200 kV associated with powdery mildew resistance, the best candidate is wheat tandem kinase. This candidate has subsequently been validated using Griggs. This work was done in collaboration with Beat Keller's group at the University of Zurich. A word about why we use the k-mer-based GWAS. If we use the traditional SNP-based GWAS for this phenotype, we detect a weaker association signal at the same locus. When we look under the peak in the reference genome used for calling SNPs, there is a 60 kb deletion compared to the resistant haplotype, which contained the wheat tandem kinase. This shows that the k-mer-based GWAS allows us to capture the large variations using multiple assemblies in a computationally efficient manner. The second trait that we looked at is leaf trichome density. Even though this trait is important for drought tolerance, it appears to be absent in wheat breeding germplasm. We identified a high confidence candidate on 4DL for this trait, which encodes alpha beta hydrolase. This work was done in collaboration with Paul Nicholson's lab at John Innes Center. Next, we looked at spikelet number variation in this panel. This trait is highly significant for improving wheat yield. Keeping everything else constant, even a 10% increase in spikelet number in wheat would be groundbreaking. We identified a high confidence candidate gene for this trait on 1D, which encodes 3-halo-6-phosphate phosphatase. A gene belonging to this family is known to play an important role in the inflorescence architecture of maize. This work was done in collaboration with Gutschall Barad and Randy Kutcher at the University of Saskatchewan. All the genes that I have described so far are only present in lineage 2 of Aegilops tauschii. The 3 lineages of Aegilops tauschii are so divergent that they rarely share any alleles. Therefore, it is generally recommended to run the GWAS separately on the different lineages. However, there are some exceptions. Wheat culm mite resistance in this panel is an example of such an exception. This trait was analyzed in Jesse Polland's lab at Kansas State University. Running GWAS using phenotype data from both the lineage 1 and lineage 2 results in a discrete peak on 6DS. When the distribution of the 300 kb associated haplotype was checked in the diversity panel, it was found to be restricted mostly to lineage 1. But interestingly, the entire 300 kb haplotype is also present in a small number of lineage 2 accessions. This indicates a reticulation event from lineage 1 to lineage 2. All the genes that I have talked about appear to be absent from the historic wheat gene pool. This represents a great opportunity to use the D-genome progenitor for trait discovery and deployment of the identified genes into wheat through transformation and introgression, or by making synthetic wheat. The sequencing data used in this study is available at Grassroots Data Repository, and the germplasm is available both at the John Innes Center and Kansas State University. I would like to acknowledge all the funding agencies and the collaborators who contributed to this project. In particular, I would like to acknowledge the members of the OPENWILD Consortium who contributed to the crowdfunding effort to generate the sequencing data for this project. Thanks, everyone.